Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So thank you so much for joining me on this highlight series. So today we're going to highlight the topic Chemiosmotic hypothesis. So it's a very important concept in photosynthesis in plants. But this is the same concept that students get really demotivated because they don't actually properly understand this chapter. So in that context, so that is the reason, or that is the main objective of bringing the entire concept today. All right, yes. So shall we talk about the Chemiosmotic hypothesis? So this hypothesis, so first of all, students have a doubt, ma'am, what is hypothesis? So then when I say there's a proton gradient created, so they understand till that much. Then when I say why the proton gradient is increased, then that's where they freeze, so they do not know anything further. So today we are going to unsolve the mystery that's we are going to solve the mystery, or we are going to crack out the puzzle that's there with us, that is your Chemiosmotic hypothesis. So don't worry, so it's going to be very easy. So I'll quickly brief you about the entire Chemiosmotic hypothesis. So it's going to be a very short lecture. So I hope you're benefiting from this highlight series. So please do like, share and subscribe. Don't forget to do that. So first of all the best for all those who are going to give your English board exam tomorrow. Yes, so here we go, highlight series, your Chemiosmotic hypothesis. So this comes in your chapter photosynthesis in higher plants. So in the previous classes, we have been talking about photosynthetic pathways, right? So we spoke about the C3 cycle, the C4 cycle, the C2 cycle, the CAM plants. So when we were talking about the C3 cycle, so we said for one molecule of carbon dioxide, so we need 3 ATP and 2 NADPH. Where and how does this ATP come from? That is the first question, right? So when I said C3 cycle is happening, carbon dioxide is getting fixed to get converted into glucose molecules, so ATP is getting utilized. So where does this ATP come? It's not that someone from above is throwing the ATP. No. How does the chloroplast synthesize ATP? So to know or to decode how chloroplast synthesizes ATP, so that is when scientists formulated the Chemiosmotic hypothesis. Now, what is a hypothesis? So when I say something is happening for sure, that becomes a process. When I am just assuming that this is how it would happen, that is a hypothesis. That is what scientists did here. All right? Okay. So what is this Chemiosmotic hypothesis? So first of all, your doubt is clarified. So why we are studying Chemiosmotic hypothesis? Because that is the way how your ATP is synthesized in the chloroplast, and your ATP and your NADH are being used in the fixation process of carbon dioxide in the C3 and the C4 cycle. Right? Now for this Chemiosmotic hypothesis to happen or to produce your ATP, so we need what is called as a proton gradient. So just like for you to enter into MBBS or have a good medical college, so you need to clear NEET. So much in the same way, at all cost, right? So now if this Chemiosmotic hypothesis has to happen, there has to be what is called as a proton gradient. So I hope you know what is a proton. So a proton is a positive H plus ion. So proton is nothing but a H plus ion. A proton gradient is across the membrane. So who is the membrane here? The membrane is the thylakoid membrane. So before we actually go into complicated terms like this, so let's first understand the structure of your chloroplast. So what is the structure of a chloroplast? So chloroplast is a double-walled organelle. So you have the outer membrane, you have the inner membrane, and you have the intermembrane space, right? So this is the outer membrane, this is the inner membrane, and this is the intermembrane space. Right? So this space inside the chloroplast is called as stroma. That is, the cytoplasm of the chloroplast is called as stroma. So inside the stroma, you have dish-shaped structures like this. So each of the dish-shaped structure is called as your thylakoids. So the center portion of the thylakoid is called as lumen. So when you have the thylakoid stacked like this, it is called as granum. So granum is singular, grana is plural. So when I have two more stacks like this, and each of them is connected via what is called as your stromal lamellae. Fine? Yes. So here for the synthesis of ATP, so my synthesis of ATP happens inside this chloroplast, right? So inside this chloroplast, the ATP synthesis happens by this hypothesis, that is chemiosmotic hypothesis, all right? So inside this chemiosmotic hypothesis, I have to have a H plus gradient or a proton gradient across the membrane. So this gradient, so first of all, gradient is difference in concentration. So one side I have to have more H plus concentration, the other side I have to have the H less plus concentration, right? So this happens across the thylakoid membrane. So inside the thylakoid, I am going to have more H plus, and in the stroma, I am going to have less H plus. So a gradient is created, up and low is created, yes. So accumulation of proton happens on the inner side of the thylakoid, okay. So now what causes this proton gradient? So what causes this differentiation? So I said inside your thylakoid, so you have more amount of H plus. So this is your chloroplast. I am only drawing one thylakoid membrane. So this is your stroma and this is your lumen. So I have to set up a H plus gradient. H plus gradient means more amount of H plus concentration, right? So there should be a difference in concentration of H plus. So on the inner side of the lumen, I have more H plus concentration, and on the outside in the stroma, I have less H plus concentration, okay. So how is that I am having more H plus concentration inside the lumen? So that is what we are going to see. The first reason why there is more H plus concentration inside the lumen is because of the splitting of water. So splitting of water happens where? In the PS2, right? So what is your, how does it happen? So here we go. So to give you a quick idea of how it happens, so we have the light. So light falls on the 𝐿𝐻𝐶 of PS2 first. So this is in P680 nanometer. So the electrons are excited. So the electrons move up. So you have the primary electron acceptor, right? So from here it comes to your ETS. So from ETS it goes to plastocyanin. Then it comes to the 𝐿𝐻𝐶 of PS1. So this is at a wavelength of P700 nanometer. So this goes up. So this here you have primary electron acceptor. Then it goes down to convert your NAD into NADPH plus H plus, right? So when it moves inside the ETS, you have the formation of ATP from ADP plus PI. Right, so this is how it happens, yes. So who replenishes the electrons? So when light falls, all the electrons will be excited, but still light is falling. I need to replace my stock. So the stock is replaced by the splitting of water molecules. So here associated with PS2, my water molecules split into H plus, electrons and oxygen. So this oxygen moves into the mitochondria for the respiration process. So this H plus is present inside the lumen and electrons are being utilized for this excitement. So as a result, H plus is happening inside the as a result, H plus is getting accumulated inside the lumen. Yes. So this is there inside the lumen of this is there on the membrane of the thylakoid, right? So this splitting, so it is like this. So this is my thylakoid membrane. Okay, so this is my stroma, right? So in my stroma, my H plus is getting produced. So as a result, more H plus is there inside my stroma. Yes. So when you look here, so this reduction also happens inside the thylakoid only. So this happens in the presence of the enzyme NADP reductase. So again H plus ion is getting accumulated. So this actually happens inside in the stromal lamellae. So this actually happens inside. So because of the splitting of water you have more H plus, because of the reduction of NADH you have more amount of H plus. Two ways, right? The third way is, if you can see, there is something called as an ETS, right? ETS is electron transport system. Just like when I say this is a student exhibition where students can come and visit, at the same time I can ask for student volunteers as well. So just like I say it's electron transport system, even electrons are required for the proper functioning of the electron transport system, right? So the electrons are needed for the proper functioning of ETS. Now where will the electrons come for the proper functioning of ETS? It comes from the stroma. So this is our this is not a stroma, this is a lumen. I am really sorry. This is our lumen. And this is our stroma. Now the electrons for the electron transport chain comes from the stroma. So the electrons are here. So the electrons are transported inside via a H plus carrier. So H plus carries the electron and moves inside. So once it moves inside, the electrons go back to the ETS. But H plus gets accumulated. So there are three ways by which H plus is getting accumulated inside the lumen. So first is by the splitting of water, the second is by the reduction of NAD, and third is by the transport of electrons. Clear? These are the ways by which your accumulation of electrons happens inside the lumen. Now whenever a gradient is made, so you have more concentration of H plus inside the lumen and less concentration of H plus in the stroma. So there is a difference that is being set up. Once the difference is set up, all that we have to do is that this has to break. This stereotype has to break. So this has more H plus, this has less H plus. So now this difference has to break. First a difference has to be formed and then the difference has to break. How this will break? So here you have a complex called as the ATP synthase complex. So this has two components, one is F0, the other is F1. So F0 is an integral membrane protein and F1 is a transmembrane protein. So when the difference is established and when they come to know that yes, there is more H plus inside than the H plus outside, the H plus will try to come out like this. Once the H plus moves through the F0, there is a conformational change in the F1. Now this F1 will convert your ADP into ATP. So your ADP is converted to ATP. Alright. Yes, so this is your chemiosmotic hypothesis. Any doubts so far? So first of all, what is chemiosmotic hypothesis? The reason why your ATP or how your ATP is synthesized inside the chloroplast. Why should the ATP be synthesized? Only then your C3 cycle and your C4 cycle will happen. Right? So how does this ATP get formed? Because of the proton gradient. What is gradient? Difference. Where is this difference? Across the thylakoid, right? On the inner layer of the inner center of the thylakoid, which is lumen, you have more H plus because of 3 lesions, and on the outer stroma you have less H plus. When this gradient is at its peak, so this gradient tries to break with the help of the ATP synthase, which has the F0 and the F1 portion, and thus your ATP is getting produced. Is it not easy? Is it not really simple to understand? Yes. So long you were pondering about this easy topic as the most tough one. Yes. So I hope it's clear. So I have it here. So first of all, you have splitting of water molecules. So when you split water molecules, the H plus is on the thylakoid interior. Though your PS1 and PS2 is on the membrane of the thylakoid, the splitting happens on the inside. So this is stroma, this is your thylakoid. So you have more H plus here, right? So primary acceptor of this is one reason. The second reason is that the electrons are accepted by the ETS. right? And who carries the ETS? So who carries the electrons for the ETS is your H plus ions, right? So when this molecule passes on its electron to the electron carrier on the inner side of the thylakoid membrane, and H plus gets accumulated on the lumen. The third reason is that the NADP reductase happens. So if okay, so this is how the H plus moves inside to transport the electrons, right? So first of all, splitting of water has happened, so you have more H plus. The second reason is that the electrons are carried with the help of your H plus ions, so your H plus ions are more, right? The third reason is that the NADP reductase happens, so this one. So NADP plus gets converted into NADPH plus H plus, right? So again you have H plus accumulation. So why this proton gradient has to be there? So when there is a gradient, the gradient has to be broken down, only then ATP can be synthesized. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane, for which you need the ATP synthase complex, which has the CF0 and the CF1. So the CF0 is like an integral membrane protein, whereas the CF0 forms a transmembrane channel, so it is in the middle, right? And your CF1 is like an integral membrane protein, you can say. So it looks like this. So F0 and F1. Sorry, this is integral membrane and this is transmembrane. So your H plus will move like this. So when it moves, the conformational change happens and your ADP gets converted into ATP. So what are the requirements for your chemiosmotic hypothesis? First of all, you need a membrane, right? That is your thylakoid membrane here. You need a proton pump, because of which there is more H plus inside one region. So the lumen has more H plus because of three reasons, that is the means by which H plus is getting accumulated. So that is your proton pump. The third one is a proton gradient. So one region has more amount of H plus, the other should have less amount of H plus, right? So there should be a pump. The last one is ATP synthase, which has two parts, CF0 and CF1, so because of which the gradient breaks and the H plus comes out, and because of which your ATP is synthesized. Alright, clear? Yes? So your chemiosmotic hypothesis has been highlighted, so I hope your photosynthesis chapter is completely covered. So there's nothing that is really tough now, because I have cleared your doubts on C3, C4, CAM, C2 and your chemiosmotic hypothesis. So tomorrow at 3:15 we have the highlight series, where we are going to start with the respiratory pathways in plants. So where I'll be elaborating about glycolysis, link reaction, Krebs cycle, and then we'll move forward to your electron transport chain. Alright, yes, so this is my schedule for tomorrow. So I hope you enjoyed my lecture today. So if you genuinely liked my way of teaching and this kind of highlight series, so please do like, share and subscribe, and please let us know your requirements and needs. So please get in touch with us through the Telegram group. The link is mentioned in the description box beneath my video. So thank you once again for watching my lecture today. So please don't forget to like, share and subscribe one last time. So, and this is me signing off till tomorrow 3:15. So I'll catch you up with another new lecture. Alright, so thank you and take care. Bye.